Welcome friends. In this video we are going to focus on biology paper 2 and this science paper is coming from the year 2021 and it was written by the internal candidates. So let's look at the first question. Figure 1.1 and 1.2 show freshly cut potato chips suspended in solution E and F at the beginning of an experiment and after 2 hours respectively. So whenever you are dealing with solutions in relation with potato chips or maybe any other food stuff, it's indirectly asking us to use the information that we have about the movement of water from one region to another region. So to summarize, we are looking at the osmosis process. Now let's take a look at these two solutions and what happened. So let's go to solution E. So the solution E, after 2 hours, we can see that the shape of the potato chip reduced in size, telling us that the cells of these potato chips lost water into the solutions. So that's what happened. Then if you look at solution F, the opposite happened. For this one, we can see that there was a gain of water because we can see that the size increased. So now, after we have this full information, one potato chip lost water, then the other potato chip gained water. Now let's see how we can put this information into answering questions. So A1, explain the difference in the appearance of the potato chips in solution E and F after 2 hours. So say the potato chips chip in solution E lost water through osmosis process. So through osmosis process, as a result, it shrinked, whereas the potato chip in solution F gained water. Through osmosis process. As a result, there was an increase in size. So this is what happened. Then if what, we can also look at these two potato chips by looking at their appearance. And it's all about their appearance. So we are saying the one that was in solution E, the appearance changed, it shrinked, it reduces in size, while the one that was in solution F, it reached what we call a turgidity where it gained water. Now we can't expect the bursting of the cells under plant cell. We know that plant cells have the cell walls. As a result, they do not burst even after gaining a large amount of water. So in summary, we are saying the one that was in solution E shrinked. So it reduced in size. Then the one that was in solution F reached what we call turgidity because it increased in size. So that is the difference. Now let's move on to Roman number 2. Based on the appearance of the potato chips in solution E after 2 hours, what term could be used to describe solution E? So we have 3 types of solutions. There is hypertonic, hypotonic, and isotonic. Now let's maybe see if we can analyze these solutions. So let's say we have a solution here where we have a plant cell. So let's just say this cell. Now we'll say the cell has 10% of salt solution, then the solution has 5%. So let's just say 5%. So whenever you are trying to find out the solution or the region where there is too much water, so in terms of concentration, where there is a lower concentration in terms of salt solution is where there is a higher concentration of water. In summary, we are saying where there is too much salt, there is less water. Where there is less salt, there is too much water. So if we have this as our illustration, we can tell that in these cells, this there is 10% and the other one has 5%. This means that the cell will gain water because there is too much water outside the cell due to the percentage concentration. So now, what type of a solution do we have when a cell gained water? So when a cell gained water, this is what we call hypotonic solution. So hypotonic solution is a solution where the concentration is less than that of a cell. Now, what if we make our illustration above the opposite? Now let's say in the cell there is 5% of salt, then outside here we have 10% of salt. So this indicates that there is too much water in the cell compared to the outside region of the cell. So water will move where there is high concentration of that. So water will move from the cell into the solution. As a result, the cell will shrink. This is what we call hypertonic solution. So whenever you put a cell in hypertonic solution, the cell loses water. If you put in hypotonic solution, the cell gains. So in summary, in hypo it gains, in hyper it loses. Now, what of a solution where we have equal concentration? The cell has 10% salt solution, then the solution has also 10%. What happens? So in this case, we are not going to have any effect because the concentration are of equal. So the water molecules move inside the cell and also at the same time move outside. So because of that movement, then we are going to have the same. So that's a picture. So if we go back to our diagram, which is 1.1. So let's look at E. E lost water. So we said whenever it loses water, then that is hypertonic solution. Then whenever it gained water, it is hypotonic solution. So what is the question? So the question is about E. So say hypertonic solution because the cell lost water. So what you can do here is you can make maybe a summary where you can just say hyper, the cell lost, hypo, the cell will gain. So that's what you can look at here. Then iso, nothing will happen. Then what would be the state of the cells in the potato chips in solution F after 2 hours? So in solution F, we are looking at the cells gained water. So what would be the state? So the state is turgidity. Turgidity, this should be. So it's turgidity. Then state the importance of the state of the cells in B1 above to plants. So because of the turgor, because the cells have gained water, we can maybe have a picture of if something gains water, the size will increase. So turgidity is important. The first one for growth, because for cells to become bigger in size, we want to gain water. So for growth, then the other one for support. So these can be the answers. Then C, explain the meaning of the term isotonic solution. So remember, isotonic solution, we said we have equal concentration. So say this is a type of solution in which the concentration is equal to that of a cell. Okay, so this is what we can share for this. So now let's move on to question 2. So question 2a, table 2 shows various food tests carried out by a learner. So most of the learners, or maybe individuals at large, people expect food tests to just be part of the practicals. So now what I'm saying is the food tests can also be just taken as also questions. So there are questions that you should expect to be given under food test. So don't just take food test as only the practicals. So like as we can see, we have been given a table that summarizes some of the food tests here. Now let's look at the first one, starch. Then the test, which solution is used to test for starch, is iodine solution. So the iodine is the solution that you need to have whenever you're dealing with a starch test. Then the color of iodine is this is a brownish color. Then the color change from brown to blue black. So the conclusion is starch is present. Then here we have emulsion. So whenever you see emulsion, it is a test for fat. Whenever it shows white precipitate, then you say starch, meaning not starch, fat is present. Then we have protein, test, burette, observation. So say color change from blue to purple. So whenever proteins are present, we have a purple. So we have answered the first question, which is complete the table by filling in the spaces. Then which nutrient in the table would cause kwashiorkor if lacking in the human body? So you know that this is if especially in children, lacking of protein may result in kwashiorkor. Then name a nutrient not in the table which helps to prevent constipation. So it's roughage. State 2 deficiencies, disease in plants. So we have leaf flaking. Also we have leaf chlorosis. Okay, so now let's move on to question 3. Figure 3 shows a longitudinal section through a root cap. Okay. So here we are looking at the cell division plus C. So we have the cell division, cell elongation, and cell specialization. And whenever you're dealing with this is the primary growth, because we are looking at the increase in terms of the height. Now identify the parts labeled G. So G, these are just the root hairs. So root hair. Now don't confuse a root hair to a root hair cell. So a root hair cell is just a cell that forms the root hairs. So that's the picture. Then our J, this part that protects the part of this plant during growth, is what we call a root cap. Okay, so now let's look at Question B. Which labeled part in figure 3 is responsible for generating new cells? So this question is same as which part do we expect to have cell division, because cell division is all about the production of new cells. So cell division is region I. This is where our cell division is. So after the cells have divided, what happens? They have to grow bigger in size. So cell elongation talks about the increase of the cells in size. Then somewhere here we may expect to have cell differentiation or cell differentiation, where the cell now becomes specific for a certain purpose, or where now the cells will have that. Now, which labeled part in figure 3 is responsible? So we know it's part I. Then we have region H is responsible for cell differentiation. Where is region H? So cell differentiated region H, okay? So cell differential. Okay, so since they have mentioned that, we take as H as for cell differential. But in between cell differential and cell division, we have cell elongation. So if cell elongation should be here, since the cell division is here, cell elongation should be here, then that's how. So the first one, cell division, cell elongation, then cell specialization. Now let's look at what the question is. Region H is responsible for cell differentiation. Apart from vesicular bundles, state 2 other cell types differentiated by region H. So apart from these, we have parenchyma. Then we have collenchyma. Then state 2 types of cells found in a vesicular bundle. So vesicular bundle has to do with the transportation. So vesicular bundle has to do with transportation. So which is we have xylem. Then the other one is phloem. Which named types of cells in C2 allows movement of substances upward and downward depending on the needs of the plant? So it's the phloem. Phloem can transport the food substances upward or downward, depends where the food is needed. Then D suggests one meristematic structure not in the diagram responsible for bringing about secondary growth in dicotyledonous plants. So this is a cambium or cambium. So we have a cambium. Okay, so we move on to question 4. Figure 4 shows the structure of a nephron. So K is bringing blood to the glomerulus and Bowman's capsule. Then our L, this is our L, which is our Bowman's capsule. Okay. Then our M, it's this vessel that carries the glomerulus filtrate until it becomes urine. Now identify the parts labeled D, K. So K, it's afferent. So the one that brings blood is afferent arteriole. Then L, this one here. So L is just the Bowman's capsule. Name 2 substances present in fluid in K but absent in the absent from the fluid in M. So this question is same as which substances that does not pass through the glomerulus hose into the Bowman's capsule. So the first one, we can look at white blood cells. inclusively red blood cells also. Also proteins if possible. Then give 2 reasons for your answer in B1. So say they are too large to pass through. To pass through the hose. Then the hose, the walls of the glomerulus are thin. Okay. State 1 common disorder associated with the kidney. So we have a kidney failure. A kidney failure is a condition or disorder where the kidney cannot work properly. Explain how the disorder in C1 can be treated. So by kidney transplant, where a healthy kidney is taken from a healthy person to someone with a disorder, or maybe by kidney machine. Then we have question 5. Figure 5 shows 4 animal cells at different stages of mitotic cell division. Okay, so cell 1, cell 2, cell 3, cell 4. Okay, now let's look at the first question. So name the structures labeled N and O. What's our N? So N is looking at this. So this structure is what we call a nuclear membrane. So if you didn't know that there is a nuclear membrane, now you know. So it's a membrane that covers the nucleus. Any what of O? Or these are chromatids. The chromatids forms the chromosomes. So we have the chromatid. So at which stages of mitotic division are cell 1 and 3? So let's go to cell 1. So cell 1, we can see that the chromatids are almost here, then the other one are here. So the cell division journey have started. Now if you want to look at the way these cell division stages are involved, you can use this to remember. So we have IPMAT. This is the order, where the first one tells you about interphase. So interphase is where the cell starts to prepare for the journey of cell division. Then after this one comes prophase. So the prophase, where the cell gets instructions from the nucleus that it is time to undergo cell division. After cell division, we have metaphase. Metaphase is where now the chromatids starts to rearrange. From there, we have anaphase. The anaphase is where now the chromatids have moved to one side, like for cell 1, that is anaphase. Then from there, we have the telophase. Then the telophase is almost the final, where we can see that cell F, the cell is almost done with cell division. So cell 1 is anaphase. Cell 3, that is the metaphase. So when answering this, you just have to look at the stages, then also the arrangement. Then C, a husband and wife were heterozygous for blood group A and B respectively. with the help of a genetic diagram, show the possible blood groups of their children. So we'll say parent. So let's say parental generation. So we have the husband, then the wife here. So now let's maybe first begin by phenotype. So phenotype, the husband, blood group A, then the wife, blood group B. So in terms of blood, whenever you look at the phenotype, you are just looking at what type of blood does that person have. Then after this, we can look at the genotype, parental genotype. So parental genotype, so genotype of P1. So how do you do this? So let's look at the statement. So whenever you have heterozygous, then we are going to have for A, heterozygous for A is just the AO. Heterozygous, there is A and O. Even for B, BO. So you can write like this, or if you want, you can use this where they use the letters I. So you can use this, you say A. So this is the heterozygous. So what I want you to focus on, don't worry about these letters, letter I. The focus is if the language is heterozygous for A, then it's AO. Heterozygous for B, this will be BO. So that's a pattern here. Then after this, what are we supposed to have? So let's look at now the gametes. So gametes of first parental generation. So which is this one? Now here we are going to have random fertilization. Random fertilization, we don't know which gene will meet with the other one. So here we are going to have a B. So there is a chance to have a B. Then there is a chance to have a O, which is A here. Okay, then here. So say we'll say genotype of F1. So there is a chance of having AB, and remember AB is just the codominance. Then AO, heterozygous, we know what AO is, so that is blood group A. BO is blood group B. Then OO is blood group O. So this is how we can answer this question. So for this video, we've just looked at section A of this paper. So that's it for this video. Thanks for watching.